Okay, this video is going to be about muscle tissue, and then the next one will be the last one. It'll just be a review for the final exam. So we'll go over muscle tissue. It's a fairly long one. I'm sure you've already looked over it, but we'll talk through it just so you can hear it lectured to you and try to pick up on different points that I may emphasize. So muscle tissue. We have some functions of muscle tissue that produce movement. Obviously, helps maintain posture, body position, helps with protection, maintaining body temperature, storing nutrients. And then we have some properties associated with muscle. So excitability, which means um, it can receive stimuli and respond to stimuli from the nervous system. Contractility, which means it can shorten. Extensibility, which means it's stretching, and then elasticity, which means it has a recoil and can go back to its original length. Okay, so just like with the nerves, we have coverings and we have levels of organization. Okay, so we have the epineurium for the nerves, we have epimysium, paramysium, endomysium. Okay, so the epimysium is epi, which is on top. So that's over all of the muscle. Okay, epimysium, and then we have paramysium around the muscle fascicles. Okay, so we have multiple muscle fascicles within the muscle. And the muscle fascicle is made up of muscle fibers. Okay, so the tissue around the muscle fiber is the paramysium, or sorry, is the endomysium. Okay, and then within each muscle fiber, we have even smaller myofibrils, which we will look at a little bit. So the way skeletal muscle is formed is very long fibers. So these are really long cells. So in order to be efficient, we have multiple nuclei. Okay? If we had one nuclei on one end of the cell and we needed to send signals or have information sent from the nucleus all the way to another end of the cell, and we had a really long muscle fiber, it'd take a really long time to get there. So we have multiple nuclei working throughout the cell to make it more efficient. So the way that is formed is we have the myoblasts. Okay, these are like the kind of like the stem cells, basically, where we have myoblasts that form together and they contribute their nuclei. Okay. And then as all of them fuse, we form multinucleated skeletal muscle fibers, which are then striated, and we'll look at why they're striated in a minute. And then the unfused myoblasts are left as myosatellite cells. Okay. So in adults are called myosatellite cells. So we'll look at the basic functional units here in the muscle. So all the way down, the tiniest portion here, these myofibrils, that's what we're looking at here. So we have myofibril here. So this is a muscle fiber and multiple myofibrils, which is here. Okay, so this part is right here. So we have a thin and thick filaments that we'll look at, but each muscle fiber has a sarcoplasm, okay, which is like the cell membrane, okay, and the or sarcolemma, and then the sarcoplasm is the like cytoplasm within the cell, okay, and then we have around each of these sarcomeres, these repeating functional units, we have the blue thing here, the sarcoplasmic reticulum, and then the connection from the sarcolemma to the sarcoplasmic reticulum here, these yellow tubes are called transverse tubules or T-tubules. Okay. And then the unit with the fused portion of the sarcoplasmic reticulum, T-tubule, and then the adjacent fused portion, this is called a triad. So we have the terminal cisterna, which is the fused portion, of the sarcoplasmic reticulum, T-tubule, and then the other terminal cisterna forms the triad here. So now we'll look into the sarcomere itself, the repeating unit. This repeating unit causes these striations. This is why it's striated, because they repeat in this way. So in the myofibril, we have actin and myosin. So actin is the thin filament, and the thick filament is myosin. Okay, so within this repeating unit, we have the myosin, which is the purple here, sorry, the purple, and then the red portion here is the actin, and then we have these little green lines. This is the portion that connects the actin with the Z line here. It helps stabilize and anchor this filament in. That's called the titin. So we'll go to the next slide here. So we have the titin, that's the elastic, that's this green. See the titin filaments here. So that attaches to the Z line to help stabilize it and keep these filaments lined up. Okay, so we'll talk about the different bands and the different labels here, so that when we look at how it shortens, we can see which bands and segments shorten and which stay the same. So when we talk about the bands of the sarcomere, we start with the A band. Okay, so the A band contains regions that overlap myosin and actin. Okay, so the region that overlaps. Okay, the M line is the center of the A band. Okay, back to this one, we can see too. This is a good look too. So we have the M line, which is the middle of that myosin. Okay, M line. So the A band is this whole section of overlap. Okay, then we see the I band is the portion where we just have the thin filament. Okay, so we just have the thin filament for the I band. The A band is the where we have the myosin and overlap between the thick and thin. So the M line is this center portion here, right in the middle of the myosin. The H band contains the thick filaments and no thin filaments. So that's just that purple section. Okay, we don't have any of the thin filaments there. And then we have the zone of overlap, okay, where the thin and thick overlap. So as we shorten this and we bring the thin filaments together and we create more overlap, this H band is going to become smaller, okay, where there's only thick filament and no thin filament. Because as we shorten these and bring these together, that's going to become smaller. Okay, the I band contains thin filaments and no thick filaments. Okay, we have the Z line. This is is in the middle of the I band, and it breaks up the adjacent sarcomeres. Okay, so that's the beginning and the end of each sarcomere is a Z line. Okay, contains the actinin, and then we have the titin fibers, which are elastic protein that extends to the Z line, and we saw that with the green, this little green wavy line. It's the titin here. Okay, so as we shorten, we're going to look at this makeup of each of these filaments here. But as we shorten, you can see that the H band gets smaller. Okay, as these thin filaments come together, this H band gets smaller. The A band gets smaller. Okay, but the zone of overlap increases. Okay, you can see the sarcomeres from the Z line here to the Z line here. So get familiar with the different labeling portions, the different segments and lines there. as we go through and understand the contraction. So let's start with the makeup of these filaments. So the thin filament, first, the actin, we have the F-actin, filamentous actin. So we have G molecules that are bound together, G-actin molecules that are bound together in these strands, okay, that kind of twist together. Okay, and these two twisted strands is the F-actin. Okay. Now, there's a line in the middle here, this little filament called nebulin, which kind of connects the two. Okay? It connects the two strands of F-actin together. And then on the F-actin, we have this yellow portion. Okay, you can see how the yellow portion is being kind of covered up by this tropomyosin. Okay, this yellow portion is the active site. This is where the myosin head is going to attach. Okay, so the active site on the F-actin. Okay, it's being covered up by the tropomyosin. And then we have little units, 3 subunits as part of this troponin. Okay, so we have one subunit that binds to the tropomyosin that locks them together, and then we have one subunit that binds to the troponin, and then we have one that binds to the G-actin molecule, and then we have one subunit that receives calcium. Okay, so we have one subunit for the tropomyosin, one subunit for the G-actin, and one subunit for the calcium. And we'll see that calcium, when that attaches, it causes the active site to be revealed. Okay, it moves this tropomyosin off the active site so the myosin head can attach. Okay, the thick filament is this purple myosin. Okay, we can see that we have many myosin heads. Okay, these things are the myosin heads. We have many of them, and that is what is going to bind to this active site when we look at that. So the thick filaments contain about 300 myosin molecules. Okay, so this one is a myosin molecule. It's got a tail and a head and a little hinge here where we see this head kind of pivot, which we'll look at that when we look into the contraction in a second. So all of these are connected and arranged around this middle line, this M line. Okay, and the H band again contains just this thick filament, and this gets smaller as the myosin heads connect to the actin and cause contraction and pulls the thin filaments together, so it'll get shorter. What also gets shorter is as these pull the thin filaments. Okay, this area where there's no thick filaments, this thin filament only, okay, where the I band is here, this gets shorter as well during contraction. Okay, okay, so those are the two filaments here. So this describes the sliding filament theory. This shows you the different bands and areas that get shorter during contraction. So as this sarcomere shortens during contraction, as the myosin head attaches to the active site on the actin and rotates and causes this contraction, okay, we see that the A band stays the same, okay, but the H band gets shorter. Okay, the A band is where we have thick and thin filaments together. Okay, that thick filament is going to stay the same length regardless, so that's going to keep that A band the same length. And then the H band, where there's just thick filaments, okay, that's going to get shorter as we pull the thin filaments together. And as we pull the thin filaments together, the area where there's only thin filaments, this I band, is going to get smaller as well around the Z line. So that's the sliding filament theory for muscle contraction. When we have a fixed one fixed side. Okay, this side is not going to move. And we have the movable side that contracts in towards this fixed side. We have freely movable sides. Both sides will move in towards each other during a contraction. So let's look at what happens during a contraction. Okay, so we have a motor neuron. Okay, we have a motor neuron that will have an action potential sent down. Okay, the action potential reaches the synapse, and all motor neurons release acetylcholine, which we've already learned. So we have the vesicles, synaptic vesicles here, releasing the acetylcholine into the synaptic cleft. As it reaches the synaptic cleft, it then finds the acetylcholine receptors. When it binds to these acetylcholine receptors, it allows sodium to rush in and can cause the propagation of this action potential down the sarcolemma of the muscle fiber. Okay. This is from your book, so it's probably a clearer picture when you actually look in your book and read that. But here's another explanation of that. So we see the synaptic vesicle. So we have the action potential coming down the motor neuron, causing the release of acetylcholine at the synaptic cleft. Acetylcholine attaches to these acetylcholine receptors and opens up the sodium channels. Okay, and these so this rush of sodium into the muscle fiber then depolarizes the sarcolemma, which causes the action potential to propagate down the sarcolemma. When we have that propagation, it then goes down a T-tubule, the transverse tubules. That action potential excitation continues down that T-tubule and activates the sarcoplasmic reticulum to release calcium. Okay, so calcium hangs out in the sarcoplasmic reticulum. When we have the action potential and excitation of the sarcolemma, it travels down the T-tubules, causes release of calcium ions into the muscle fiber, causing the calcium to attach to the troponin. Remember, the troponin has the 3 subunits. One of them attaches to calcium, and when calcium attaches, the troponins also attached to the tropomyosin. So it pulls that tropomyosin, rotates it off the active site. Okay, you see it now exposes this active site. And once this active site is exposed, the myosin head automatically attaches to the active site. And we'll go to this one. So this one, we see the calcium coming and attaching to the troponin, moving the tropomyosin. Okay, once it moves, the myosin head forms an attachment with the active site. This is called a cross bridge. Okay, that's cross bridging. So when this cross bridge forms, it's when the myosin head attaches to the active site of the actin. Then the myosin head goes through, it kind of pivots at this hinge. Okay, this pivot is called a power stroke. Okay, this is what causes this shortening. Okay, when this pivots, it pulls this thin filament closer together. Okay, pulls the thin filament over the thick filament, the sliding filament theory. Okay, so we have multiple myosin heads attaching, going through a power stroke, and drawing the thin filaments closer together. Okay, now in order to release from the active site, we need ATP. We need energy to bind to this myosin head. Okay, so we need ATP binding to the myosin head to release this. And then the ATP is broken down into ADP again, and then it's allowed to have another power stroke if the form another cross bridge and and pivot again if the active site is open. Okay. So if the active site is still open, it can form a cross bridge again and shorten and continue to shorten, but it needs ATP to release out of that power stroke. Okay, when we don't have ATP, like when we're dead, we don't have ATP releasing these. Okay, we have these myosin heads that shorten and stay. This is when we get rigor mortis, because we can't release the muscle. We can't release the myosin heads because we don't have ATP. So ATP energy is required to release that, to allow that myosin head to attach again to another active site, portion of the active site. So there we go. So we have, this is a muscle contraction again. So we have the action potential, or action potential coming down the motor neuron, releasing acetylcholine. Acetylcholine attaches to the acetylcholine receptor. Sodium rushes in, depolarizing the sarcolemma. That action potential continues down the T-tubule and excites the sarcoplasmic reticulum to release calcium. Calcium binds the troponin, which rotates the tropomyosin off the active site. So then the myosin head can attach to the active site, causing a cross bridge during the attachment, and then it pivots to form a power stroke to shorten and slide the thin filament over the thick filament. Okay. Then when we need to stop muscle contraction, we have a decrease in action potential. So the acetylcholine goes away, sodium channels close back up. The sarcolemma is no longer depolarized. Okay, so all the calcium then starts to go back into the sarcoplasmic reticulum, and since we don't have any calcium, the troponin causes the tropomyosin to rotate back over the active site and blocks the active site, and that causes a relaxation and release of the thin filament to slide back to a normal resting length of the sarcomere. Okay. So that's contraction and relaxation. Now, we have tension built up in our muscles when we contract our muscles. Okay, during the cross bridging, when the thin filaments slide over the thick filaments, that's developing tension in the muscle. And there's a way to develop optimal tension, and then there's ways in which we have less optimal tension. Okay, so when the shortening distance is too small, so say if we're trying to flex our bicep and our bicep is already flexed and shortened, the sarcomere is shortened quite a bit. We have too much overlap between the thick and thin filaments. We can't develop enough tension. So that's down here, we see it's completely overlapped. Even here, there's a lot of overlap, so it's not as optimal tension until we get up here where we have optimal overlap, where only overlap is where the myosin heads are. The overlap occurs only where the myosin heads are, and all the myosin heads overlap, so we can form maximum tension when we contract the sarcomere. Then we have, tension starts to die off when we have less overlap. So some of these myosin heads don't overlap the actin, so those aren't being utilized to create a cross bridge and create muscle contraction. So we only have a few of these myosin heads, so it creates less tension. Okay, so that's what this slide is showing, how optimal overlap creates optimal maximum tension. Okay. And that's what the length tension relationship. This is when tension produced by the entire muscle fibers related to the length of the individual sarcomere. Okay, so a twitch. When we talk about a twitch, slow twitch, fast twitch. Twitch is a single stimulus contraction, single stimulus from the nervous system, stimulus of the muscle fiber, contraction of the muscle fiber, and then a full relaxation of the muscle fiber. Okay, so those three parts occur during one twitch. Okay, so we have a latent period where the stimulus begins, and then we have the contraction phase, and then the relaxation phase. Now, depending on the some muscle fibers develop faster tension. Okay, so these are faster twitch fibers. Like in the eye, we develop tension very quickly, and then we have a short contraction phase and a quick relaxation phase, where we have larger muscles, okay, and more muscle fibers are innervated by less motor neurons. Okay, we have larger motor units. The tension takes a longer time to develop and then a longer time to decrease. Okay, so we have a longer twitch, slower twitch. Okay. We can we can build up increases in tension in a couple different ways. So the first way is called a TRAPE. The TRAPE is an increase in peak tension. We have a full twitch phase. So we have that stimulus, contraction, and then full relaxation. And each time we have a new stimulus, it increases the tension to the max tension load, okay, in a TRAPE. So this has the full twitch of the stimulus, contraction, full relaxation. And then we can have a wave summation where we have the stimulus, the contraction, and then not a full relaxation, okay, before we have a new stimulus. And so it kind of comes in a wave and increases tension. And then we can have what's called tetany. So we can have incomplete tetany. So this is where we don't quite reach maximum tension, okay, but we have less time in this relaxation phase. So the stimulus occurs more frequently, very rapidly, and maximizes the tension. There's some relaxation, so it can't get to maximum tension in tetanus, but it has a very short relaxation phase. And then with complete tetanus, we have no relaxation phase, just complete stimulation until we reach maximum tension load. Okay, we talked about, I just mentioned a motor unit. A motor unit is the motor neuron plus all the muscle fibers. So with things like the eye, like I said, it has 4 to 6 muscle fibers. Okay, so we can develop tension fairly quickly and have a fast twitch, and then a quick decrease in relaxation phase. But in muscles like in the hips and thighs, we can have thousands of muscle fibers in a motor unit. Okay. Fasciculation, this is involuntary muscle twitches, and it usually involves more than one muscle fiber. Recruitment, this is where we have a smooth, steady increase in the number of motor units that are active. So we slowly increase the nerves, stimulating all of the muscle fibers to develop, to recruit all of the muscle to work. Okay? And when we have a sustained contraction, say we're trying to hold a box and not drop it, we have sustained contraction of all those motor units. Our body isn't going to utilize all of the motor units all at once so that they all fatigue at one point. It utilizes an asynchronous motor unit summation. So that's where we have some motor units active at one time and other units not. So we have one motor unit 1 and 2 kind of overlapping at one time. And then as motor unit 1 kind of shuts off, motor unit 3 activates. So we can maintain a fairly steady tension within the muscle to maintain that sustained contraction. And then muscle tone, this is what our resting tension on our skeletal muscle is. And so when we have greater tone, we have higher resting metabolism. Okay, let's talk about muscle contraction. So when we, there's a couple different types of contraction. There's one where we contract and move the load closer. This is called a concentric contraction. Okay, we have an isotonic concentric contraction. Okay, isometric, the same length, same length is where we have contraction, but we don't have movement of the load. Okay, so the tension doesn't surpass the amount of the load. So the load doesn't move, but the muscle fibers are active. Okay, so we have, when we move the load, when the amount of the load is less than the muscle activation, and we move the load, we shorten the muscle. Okay, that's isotonic concentric contraction. When we activate the muscle, but do not change the length of the muscle, we can't move the load, it is isometric contraction. And then when the load becomes too much for the muscle fibers, but we're still actively trying to create tension and contraction within the muscle lengthens with contraction. So this is an isotonic eccentric contraction. Eccentric, so it's lengthening as it is contracted. Okay. This is kind of like this is what happens if you're like running downhill. Okay, your quadriceps they're contracting to slow your descent, but they're lengthening as you're going down the hill, right? So we have eccentric contraction, where we have the load causing a lengthening in the muscle as it's contracted. Okay, so go over those terms, it'll be on the test. This is a simple load speed comparison. So when we have a small load, we can increase tension and increase shortening fairly quickly. When we have a larger load, we need to recruit more, so it takes longer to recruit enough motor units to begin shortening, and the shortening is less due to the size of the load. Okay. All right, so we're going to talk about muscle metabolism here. So we need a couple of things for muscles to function normally. We need normal pH, we need blood and oxygen, and then we need energy reserves. Okay. So the muscle works off ATP, okay, exclusively. Okay, ATP is the only energy source used with muscle contraction. So we need to generate that, and we generate it 3 different ways: direct phosphorylation by creatine phosphate. We have anaerobic, so anaerobic is no oxygen, and this is called glycolysis. And then we have aerobic, which is oxygen. Okay, this is done by the Krebs citric acid cycle and electron transport chain to create ATP. So let's look at those. So we have here we have glycolysis. We're going to focus mainly on glycolysis, which is anaerobic breakdown of glucose, and then aerobic metabolism, okay, which is the citric acid cycle, the Krebs cycle, and the electron transport chain. So we're not going to go explicitly into each of those, but just so that you know the names associated with those. You don't need to memorize all of the little intermediates and things like that. But glycolysis, this is for when we don't have oxygen, okay, when we need fast energy, the demands are high, okay, when we produce ATP, it is not an efficient production, okay, when we break down glycogen into glucose. Okay, glycogen is, again, multiple glucose chains, so we break that down into one glucose, and glucose is then creates a pyruvate, which allows for 2 ATP. So 2 pyruvate yields 2 ATP. So not a highly effective system. And then we also develop a bunch of lactate and hydrogen atoms, so lactic acid builds up in our muscle too. So not an efficient system, but this is what occurs when we don't have oxygen. And we'll look at the specific muscle fibers in a minute that use mainly glycolysis. And then most of the other muscle fibers use aerobic metabolism. Okay, so this is where we have oxygen. Okay, we have mitochondria. So we have our glucose and we turn it into pyruvic acid. We use that pyruvate in the mitochondria with oxygen, and the electron transport chain creates a lot more ATP. So we get, for every pyruvate, we get 15 ATP. So we have a lot more efficient system when we do that. But again, it takes longer to get this. It's a longer cycle to get this. Okay. So when we have different vocab here associated with energy use and metabolism, so our recovery period. So this is where we have activation of the muscle, and then we have a period where the muscle fiber returns to normal. So this period in which the contraction stops, the energy need goes down, we return to normal state. This is the recovery period. Oxygen debt or excess post-exercise oxygen consumption. This is the amount of oxygen required to come back to normal state, normal pre-exertion state. Okay. Muscle contraction this metabolism produces a bunch of heat. Okay, that's what muscles are responsible for maintaining normal body temperature. Okay, so that's occurring when we use our muscles. Okay, we use our muscles, we create a bunch of heat. 58% of the energy produced escapes as heat. Okay. Okay, and the different muscle fiber types, fast fibers here. We have fast fibers. These produce, these are large fibers. They produce fast, powerful contractions, and these use glycolysis. Okay, so we don't have a lot of mitochondria within these. Okay, the reserves, glycogen reserves are large in this one. We use glycolysis, and they fatigue very rapidly. Again, because it's so inefficient in producing ATP, we can't maintain that for a long time. Plus, we have a lot of buildup of lactic acid, which causes more fatigue. So fast fibers, we have large diameter fibers that produce powerful contractions for a short period of time. Okay, so this is like sprinting. Okay, you need powerful contractions to go fast quickly, and then you fatigue. Okay, so slow fibers are about half as big. They take 3 times as long to reach peak tension. There's a lot of mitochondria because this is mainly utilizes aerobic metabolism. We also have myoglobin. So we have hemoglobin in the blood, which transports oxygen. In the muscle, we have myoglobin. Okay, this is red oxygen-carrying pigment in the muscle. So the slow fibers are darker because they have this red pigment. This is where we get our dark meat from, okay, as opposed to our white meat. Our white meat is our fast fibers, and our slow fibers are our dark meat. So this is more like an endurance runner. Okay, we have we utilize our oxygen. We go slower. We don't need as much power as fast. So that's the slow fiber. Intermediate is kind of in the middle. Okay, we have low myoglobin, but we have a lot more mitochondria, more blood flow. So it's a little bit more resistant to fatigue. It's kind of in the middle. Okay, so fast, slow, and intermediate fibers. And here's a chart if you want to go over different differences there. All right, so then we talk about the rest of the muscle tissue. So we have skeletal muscle, then we also have cardiac muscle and smooth muscle. So we got to talk about cardiac muscle. This is relatively small muscles. We have typically a single centrally placed nucleus. They're branched. They're connected through intercalated discs, which the intercalated discs are bound together by gap junctions and desmosomes. Okay, desmosomes and gap junctions formed between the cells forms this intercalated disc. These typically rely on aerobic metabolism. So we have lots of mitochondria and myoglobin. With the cardiac tissue, it is a little bit different, has different characteristics. So we have functional syncytium. So this is where we have the cardiac muscle cells and the muscle fibers act as one big muscle cell. Okay, they synchronize into one large muscle cell, basically. So they all act together, basically. We have automaticity. So this is where we have the contraction without neural stimulation. So we have pacemaker cells. This creates a regular determined timing of contractions. This sets off the contractions within the cardiac muscle tissue. The nervous system can influence the pace, so we can increase and decrease our heart rates, but we have the pacemaker cells to stimulate and activate the specific muscle fibers. They have a longer refractory period and do not readily fatigue, so that's good. We need our heart to keep beating without fatiguing. It's more responsive to extracellular calcium and cannot undergo wave summation or tetany, which is good. We don't want our heart to stay contracted. That would be bad. So here's a picture of this. So we can see the cells, the nucleus here, these intercalated discs in between. So these are the desmosomes and gap junctions. Okay, so they can all communicate with these gap junctions and act as one muscle cell. And then you can see that parts of it are branched. It's kind of hard to see in this one, but it's branched as well. But we do still see the striations because we still have these sarcomeres within the cardiac muscle. You can see it's still fairly similar to the skeletal muscle, where we have the sarcolemma, the T-tubules. We don't have the triads because the sarcoplasmic reticulum does not fuse here near the T-tubules. There's more influence of calcium, extracellular calcium on the muscle fibers. So it's a little bit different there, but fairly similar with the striations and with the sarcomeres for that. Smooth muscle is a bit different. Okay, these are formed in sheets or bundles or sheaths. These are not striated. Okay, they do not have myofibrils and sarcomeres. Okay, so these smooth muscle is usually around like vessels. Okay, so they can coordinate movement of substances through internal passageways, so around blood vessels, around the bronchi, or the respiratory pathways, around the digestive tract to control sphincters and peristaltic movements, in the bladder, in the urinary system. Okay, so these are structured differently. They're spindle-shaped with the one nucleus in the center. We don't have T-tubules, sarcoplasmic reticulums, sarcomeres. Okay, none of that is the same. We do have some thick filaments scattered throughout the sarcoplasm. We have more myosin heads, and then we have the thin filaments which are attached to the dense bodies. So we have the dense bodies are these little circles here. And then we have intermediate filaments made up of desmin that kind of connect through here. Okay, we have the myosin. You see here the purple and the actin, which is kind of the red stuff. And then the dense bodies with the intermediate filaments. Okay. So as the muscle contracts, okay, as the myosin heads act on the actin, the cell basically corkscrews shorter. Okay? It doesn't just, slide. We don't have that sliding filament theory with like the skeletal muscle. This one instead just kind of twists and corkscrews tighter. So we have a different mode of contraction since we don't have these sarcomeres. So the calcium, the cytoplasmic calcium causes contraction. So it binds to calmodulin. And the when that's bound to calmodulin, it activates myosin light chain kinase. Okay, that enzyme causes myosin heads to attach to the actin. Okay, so calcium binds to calmodulin. Calmodulin stimulates myosin light chain kinase. The kinase then causes myosin heads to attach to the actin. Okay. Plasticity means that smooth muscles can contract over a wide range of lengths and can undergo sustained contractions. We have a couple of different ways that they actually contract. So we have multi-unit smooth muscle cells. So like in arteries, in our iris, okay, we have motor units similar to the skeletal muscle, where the smooth muscle cells are connected to multiple motor neurons and they are stimulated to contract. Then we have visceral smooth muscle. So this is where we lack motor neurons. In our digestive tract, they're connected through gap junctions, kind of like the intercalated discs where they have the gap junctions. There's pace setter cells instead of the pacemaker cells within the cardiac tissue. The pace setter cells spontaneously trigger contractions of these muscle sheets, and this stimulation goes through these gap junctions, so the entire sheet or layer contracts, and this can help form rhythmic cyclic activity. So what we need for like peristaltic movement to move our food through our intestines, things like that. So 2 ways for contracting with a motor neuron through the multi-unit smooth muscle cells, and then visceral smooth muscle cells, which do not have a motor neuron. Okay. So, and then there's last one here is skeletal muscle, cardiac muscle, smooth muscle, in comparison to all of those to get an idea of that. So that was a fairly long presentation on the cardiac muscles. I won't go through the other skeletal muscle PowerPoint. I'm not going to read you origin, insertion, action. That's like me just reading charts to you. You probably won't sit and listen to that. But one thing I wanted to go over was the origin and insertion, which is important. Some of you already turned in the extra credit, which is good. So you probably already understand origin and insertion. But origin obviously is where the muscle originates. Okay, it originates at the stable location. So the part that is not moving. So if we're talking about our bicep, okay, that attaches up into our shoulder. Okay, so our shoulder is not moving. It flexes. The bicep flexes at the shoulder and at the elbow. So if we're moving, say we're flexing our elbow, okay, our shoulder is not moving down towards our forearm is moving up. So the portion that's not moving, the shoulder, is the origin, okay, and then the insertion is at the point that's actually moving, okay. So make sure when you're doing origin and insertion, we're looking at the portion that is actually moving as the insertion point. So on the test, you're going to have to, you won't I don't think we'll need origin insertion too much, but more of action. So origin insertion is going to help. So if you know that biceps brachii goes up into the shoulder and helps to flex, it crosses two joints, so across the elbow and across the shoulder. So it helps to flex the shoulder and flex at the elbow. Then you'll know that that's the action of the action of the muscle, as opposed to the brachialis, which doesn't cross the shoulder, so that just flexes the elbow. So you'll need to know more specifically actions of different muscles, and I'll talk about that in the next one, where we go over a review really quick for the final exam.